Before we proceed discussing the fourth type of macromolecule, which is nucleic acids, let's have first a short review on carbohydrates, lipids, and proteins. 10 seconds will be allotted for every question. I would like you to answer it before the answer is shown on the screen. Good luck! For this lesson, we will focus on nucleic acids. And at the end of this discussion, you are expected to first describe the structure and types of nucleic acids. Second, explain the roles and functions of DNA and RNA. Third, discuss the central dogma of molecular biology. And last, illustrate the complementary strands both for DNA and RNA. We already have discussed the first three types of macromolecules in the previous videos. These are carbohydrates, lipids, and proteins. Carbohydrates are composed of carbon, hydrogen, and oxygen. Lipids consist of CH and a little amount of oxygen, while proteins have proportional composition of CHO plus nitrogen. Putting together the various monomers of monosaccharides to form a disaccharides or a more complex polymer is made possible by glycosidic bonds. Ester bond naman for lipids and peptide or amide bond naman for proteins. The fourth macromolecule or biological molecule is the nucleic acids na binubuo ng monomers na tinatawag na nucleotides that are joined by phosphodiester linkages. Okay, this time, let's tackle more about nucleic acids. A nucleotide is the basic building block of nucleic acids. Binubuo ito ng pentose sugar molecule, which is a monosaccharide with 5 carbon atoms, either ribose and RNA or deoxyribose and DNA, na naka-attach sa phosphate group and a nitrogen-containing base. Nitrogen base or nucleobases are divided into two, the purines and pyrimidines. By looking at the structure of the two, we can say that purines are much bigger because they have a 2-ring structure or 2 carbon-nitrogen ring bases, while pyrimidines only have a single ring or 1 carbon-nitrogen ring base. The purines contain 1 hexose and 1 pentose ring, while the pyrimidine contains a single hexocyclic ring. However, purines and pyrimidines both are made up of the aromatic ring having carbon and nitrogen in it. The purines in DNA are adenine and guanine, the same as in RNA. The pyrimidines in DNA are cytosine and thymine. In RNA, they are cytosine and uracil. And if these nucleotides are assembled in a linear fashion by the formation of phosphodiester linkages, polynucleotides are formed. A polynucleotide, therefore, is a chain of nucleotides. It's a sequence of nucleotides. We do have two types. We have RNA, or just a sequence of polynucleotide, and DNA, or a pair of polynucleotides. Pero paano ito nabubuo? Okay, to find it out, let's compare and contrast first the DNA or deoxyribonucleic acids and RNA or ribonucleic acids in 5 terms. First, the structure. The most obvious difference is that RNA is single-stranded na nabuo from short chain of nucleotides, while DNA is a double-stranded molecule na nabuo dahil sa 2 complementary strands ng long chains of nucleotides. DNA has a double helix structure because hydrogen bond holds together the base pairs in the middle. Second, the types of sugar present. The pentose sugar in DNA is called deoxyribose, and in RNA, the sugar is ribose. The difference between the sugars is the presence of the hydroxyl group on the second carbon of the ribose and its absence on the second carbon of the deoxyribose. RNA contains the sugar ribose, while DNA contains the slightly different sugar, deoxyribose, or a type of ribose that lacks 1 oxygen atom. In other words, deoxyribose has 1 less oxygen molecule than ribose. Third, the nitrogenous or nucleobases. Another difference is that DNA and RNA also have nearly identical nitrogenous bases. Both have the bases adenine, cytosine, and guanine. However, DNA uses a 4th base called thymine. RNA's 4th base is uracil. Fourth, the nitrogenous base pairing. Just remember GCAT for DNA, or guanine pairs up with cytosine and adenine pairs up with thymine always. While in RNA, tandaan ang mnemonics na GCAL, kung saan ang thymine is replaced or napalitan lang ng uracil. Complementary base pairing is the standard arrangement of bases in nucleotides in relation to their opposite pairing, such as thymine being paired with adenine and cytosine paired with guanine. Using these nucleobases, let's get the complementary strands for both DNA and RNA. If we have a DNA strand of ATGC, TTCGA, what would be its complementary strand? Okay, it should be TACGAAGCT. For the second one, let's say we have this DNA strand. What would its complementary strand for RNA? Correct. It should be AUGCU-UCGA because thymine is being replaced by uracil in RNA. And last, their location or structure of cells where both DNA and RNA are commonly found. In prokaryotes, DNA is found in the nucleus, mitochondria, and chloroplast, while in prokaryotes, ito ay matatagpuan in the cytoplasm in the form of a nucleoid. The nucleoid contains all or most of the genetic material of a prokaryote. To know the difference between prokaryotes and eukaryotes, kindly check the video lesson by clicking this link. Meanwhile, RNA forms in the nucleolus and then moves to specialized liquid regions of the cytoplasm called cytosol. It is the water-based solution in which organelles, proteins, and other cell structures float. But overall, RNA can be found in the nucleus, nucleolus, ribosomes, mitochondria, and cytosols of the cytoplasm. And before we discuss the two most important functions of DNA and RNA, alamin muna natin ang correct sequence starting from nucleotides. Ang nucleotides ay ang fundamental monomers of nucleic acids, and when a long chain of nucleotides are linked together by phosphodiester linkages, polynucleotides ang nabubuo. Polynucleotides that are grouped into two, RNA if single polynucleotide chain, and DNA naman if two long chains of nucleotides are linked by hydrogen bonding. Kapag pinagsama-sama naman ang DNA, genes ang nafuform. A gene is the basic physical and functional unit of heredity. It is actually a segment of DNA that has two variations na tinatawag nating alleles. Genetic variations are the differences in DNA segments or genes between individuals, and each variation of a gene ay tinatawag nga natin na allele. Maaari itong dominant allele, which is expressed or physically manifested, while recessive allele, which is not expressed or masked. And we have actually a separate discussion on this topic, so don't forget to subscribe to get yourself posted. Okay, going back. From nucleotides to polynucleotides to DNA to genes, genes are found on tiny spaghetti-like structures called chromosomes, which are made up of two identical sister chromatids. And chromosomes are found inside cells, particularly in the nucleus. Now, i-discuss natin ang two major functions of nucleic acids. Una, for transmission of inherent characters from one generation to the next, or for storing and expressing our genetic information. And DNA is the one responsible for that. Pangalawa, for protein synthesis in a cell, performed hand in hand by both DNA and RNA. RNA comes in different molecular forms that participate in protein synthesis. Kung ano-ano ang mga forms na yon, yan ang didiscuss natin mamaya. As mentioned, protein synthesis is one of the major functions of nucleic acids. Such process or functions is described and illustrated in the central dogma of molecular biology, which shows the flow of genetic information in cells from DNA to messenger RNA or mRNA to protein. It has three processes involved. First process, from existing DNA to make new DNA, that's DNA replication. DNA replication is the process by which a double-stranded molecule or DNA molecule is copied to produce two identical DNA molecules. It is briefly explained in three steps. Unang step, DNA unwinds from the histones. Ang histones ay nuclear proteins responsible for loosening the tightly wound DNA structure. Pangalawang step, unzip the double helix structure of the DNA molecule into two separate strands. This unzipping or unwinding is carried out by an enzyme called DNA helicase na nagbi-break sa hydrogen bonding between the complementary bases ng DNA. Pangatlong step, the DNA polymerase produces a complementary strand using the sequence of nitrogenous bases of that strand. Then the last step would, with the help of DNA ligase that connects these partial strands into a new continuous segment of DNA. Second process, from DNA to make new RNA, that's transcription. Transcription is the process of creating a complementary RNA copy of a sequence of the given DNA template. Remember, DNA cannot leave the nucleus, pero ang mRNA pwede. Messenger RNA or mRNA molecules carry the coding sequences for protein synthesis. That coding sequence ay tinatawag na transcript. That's why it is called transcription. Sa madaling sabi, ang mRNA ang may dala na instructions from the genes to produce or synthesize proteins. An enzyme called RNA polymerase opens up a section of DNA and assembles a strand of mRNA by reading the sequence of bases on one of the strands of DNA. If there's an A on the DNA, there will be a U on the RNA and vice versa. If there's a C on the DNA, there will be a G on the RNA hanggang sa makumpleto ang complementary strand for mRNA. Last and third process, from RNA to make new proteins, that's called translation. Translation is the process by which mRNA is decoded and translated to produce a polypeptide sequence na tinatawag nating protein. It also undergoes three steps. Unang step, the initiation phase. During initiation, the strand of mRNA forms a loop and a small ribosomal subunit hooks onto it and finds a sequence of bases that signals it to begin transcription. This is called the start codon or AUG. The start codon marks the site at which the translation into protein sequence begins. Pangalawang step, the elongation phase. In the elongation phase, the fully assembled ribosome starts to slide along the mRNA. At pangatlong step, the termination phase. In the termination phase, upon reaching the stop codon, the tRNA or transfer RNA that binds to the first position carries a protein called a release factor. The stop codon marks actually the site at which translation process ends. To make the protein synthesis a success, three RNA structures work together. These are the mRNA or messenger RNA, the rRNA or the ribosomal RNA, and tRNA or transfer RNA. The mRNA carries the coding instructions of an amino acid sequence of a protein, while rRNA is associated with proteins to form ribosomes, and tRNA carries specific amino acids to the ribosome to finally form the polypeptide chain or protein. The main function of tRNA is to transfer a free amino acid from the cytoplasm to a ribosome, where it is attached to the growing polypeptide chain. tRNAs continue to add more amino acids to the growing end of the polypeptide chain until they reach a stop codon on the mRNA. The ribosome then releases the completed protein into the cell. This time, our last topic is the nucleic acid structure. Again, nucleic acid are long chain-like molecules composed of a series of nearly identical building blocks called nucleotides. And when the sequence of these nucleotides is linked by phosphodiester, the primary structure is formed. As you can see, the 3-OH or hydroxyl group of the sugar in one nucleotide forms an ester bond to the phosphate group on the 5th carbon ring of the sugar of the next nucleotide. And it is then linked by phosphodiester to make a long chain of nucleotides. For secondary structure, particularly in DNA, there are 2 strands of nucleotides that wind together in a double helix where the bases are arranged in stair-like steps held together by hydrogen bonding. In tertiary structure, or the 3D arrangement of all atoms of nucleic acids, is termed as supercoiling, or the further coiling and twisting of DNA helix. It can be relaxed or supercoiled. Lastly, quaternary structure refers to a higher level of organization of nucleic acids and the interactions of nucleic acids with other molecules. Alright, we're done discussing the basics of nucleic acids, but before you take the test, please don't forget to hit the like and subscribe buttons for more lesson updates. Okay, let's find out how much you have learned from this lesson. For exercise 1, you have 10 seconds to answer each question and 1 minute naman for exercises 2. And to know your result, your score, kindly click on the link provided in the video description. Good luck!